Okay, so I think the last thing we were going to do is just do a quick wrap up of the day, just make sure summarize basically, hopefully what people learned today, and then maybe give you a little preview of what we'll be doing tomorrow. And so, today we pretty much spent the day just doing some of the basics that you can do in our resources. Um, including just the ability to run a site search, with text and words, looking at a gene page. Hopefully, you got to appreciate that gene pages are full of various sections, and each section has a lot of data. And then you can open them up and toggle them and move around. And in fact, in gene pages, you can even run certain analyses like a multiple sequence alignment, or navigate to the orthology database where you can look at orthology not only within your organism of interest or your species of interest, but moving out across the tree of life. Um, and then we spent the last hour or so, or less than an hour, building search strategies and trying to learn how to leverage the various data in a particular database to answer questions. And what I was telling my group is that, and I'm sure you probably heard this in other groups, is usually you come to these databases with a question. You have a question you're trying to answer, and sometimes the question is very straightforward. It's basically, I just want to find the genes that have a signal peptide, right? There's one search, you need to find that. Um, in other cases, it's a little bit more, you know, complex, or it's a bigger question. Like, I want to define vaccine candidates for Plasmodium, or pick your pathogen of interest, or for Candida or something. Um, and and of course, we don't have a search that's just find me all vaccines, right, or possible vaccine candidates, right? That would be very nice, and maybe with AI in a few years we will be able to do that. But for right now, you kind of have to be your own. You have to be your own AI, and you have to basically take this big question, this big find me vaccine candidates, and break it down into smaller questions, right? So, for one way I like to tell people always is, well, if you're trying to find a vaccine candidate, what are the characteristics of that vaccine candidate, of the ideal vaccine candidate? And you may come up with things like, oh, it needs to be secreted, or it needs to have very few polymorphisms, or maybe it needs to have many polymorphisms. Maybe it needs to be expressed at a particular stage of the organism that you're interested in. And so very quickly you start building a list of questions, right? That are sub questions of your main big question. And now the next step is to figure out, well, can I answer these questions using some of the searches in the database, right? So if you think that all possible vaccine candidates should have a signal peptide, well, can you find genes with a signal peptide? And the answer is yes, there's a search that allows you to find signal peptide containing genes. Now you can start building your strategy and say, okay, now that I've defined all the genes that have signal peptides, I could ask, well, how many of them have the type of polymorphisms that I'm interested in, right? And so you can intersect with that, and then you can continue building this until you've answered, hopefully, most of the questions that you had, your sub questions. And then ultimately, what I've told the group I was in as well, and is that you should always take a look at the results that you're getting, and look at them and ask yourself, well, do these make sense? Like, if you were looking for a bunch of vaccine candidates and you get a bunch of things that are clearly not going to be good vaccine candidates, what happened here? Is there a mistake in the database? Is there a bug somewhere that's causing an issue? Or am I or did I do something wrong? Maybe didn't ask the right question, or maybe I need to modify one of the questions. And those are things that you can, always look at the results and ask yourself if they're correct. Like, for example, if you ran a search for um, all phosphatases in Plasmodium falciparum 3D7, and you get 6,000, right? Almost the entire genome, you should immediately think something is wrong, right? Either the database is messed up, or I did something wrong, right? And it's somewhere in between, or there, and you can figure it out, right? And so that's an extreme, obviously, but that's what I would encourage people to do. Any database you're looking at, don't just trust the results. Look at them and view it as a real experiment, as if you're looking at a Western in the lab, if you still do Westerns these days, or whatever experiment you're doing, and see if you actually if you have controls in there, do you have some positives? Do you have some negatives? Can you actually figure out? Can you build some confidence in the results that you got? I know others may have some comments as well, but before I turn over to other comments, and of course the open discussion section if people want to stay on, is just quickly as a prelude for tomorrow, we're gonna spend some time looking at the genome browser, and in particular trying to view data in a browsing manner. So instead of running searches, we're going to look at data, visualize the various experiments that we can use to look at genes and gene models, and then also try and figure out if we can leverage the data, the RNA-seq data in particular, but there are many other data types that will be very useful for this, to assess the gene models that we have. And so in the case, at least for genes that have introns and exons, this becomes kind of interesting to be able to look at the intron exon boundaries and so forth, and so that'll be potentially exciting. And we'll wrap up the day with a couple of demos. One is looking at how you can actually modify gene models in a system we call Apollo. We didn't develop this; it's developed by another group, but we gladly use it. And then also we'll talk about how you can upload results from your own experiments into UPathDB, so you can run your own searches on your data actually within the context of your private environment. So with that, this is all I was going to say. I'll turn it over to any of the other instructors if you have anything else to add. And as we said in the morning, this officially ends today's the first day of the workshop. We will stay on until around 1:30 or so for additional discussion. If anybody wants to stay on, and if you have additional questions, whether it is about the workshop itself, or you have general questions about ViewPathDB resources and the future, we're happy to answer those as well. And so you're welcome to stay on. Otherwise, if you feel free to also leave, and we hope to see you tomorrow morning. Thank you, everybody.
Okay, thank you very much. Nice talk.
See you, Alejandro.
Any questions
See you.
or comments?
See you tomorrow.
See you tomorrow.
Tomorrow.
Yeah, thank you. I have a question. Okay,
Please
just
go ahead, Stephen.
two questions. So my first question is, how can I download the table I get for my search result as it is? Because when I click download, I think the option is different criteria, pre-selected criteria are given, like gene models or transcript. But I want the search strategy, the results table that comes out. Yes, I want it. I just want it like that. I don't want the gene ID or any other.
Yeah,
First
so let me
question.
show you how you can do this. This is, of course, a different strategy. This is a strategy we were working on in my breakout group and revising from the strategy that was given in your exercise. But you can download information here, and you have a variety of ways to be able to configure that configuration. So we may have looked at the FASTA downloads to identify the sequence from the upstream sequence from those genes that were identified as having SNPs, but you might want to download other information as well. You might want to download information from the genes, choosing your own columns that you'd like to download. So by clicking on this link here, I can
David, oh, sorry, I see it. I see it now. Sorry, I didn't see your shared screen, but I see it now. Apologies.
Yeah, sorry. I assume other people are seeing the shared screen. I have chosen to download these in a tab or comma delimited file, and the columns that were shown on my query results here, which will come up momentarily, right? The columns that were shown in my query results will be what comes up by default. And remember, as Omar showed you earlier, you can choose to add other columns you might be interested in. Ortholog groups, abundance of SNPs, whatever other information. So this is what I've chosen to add, but I might want to add other information if I was interested in the ortholog groups that were involved. I could add this. And because there were only a few genes here, I'm just going to show these results in the browser. And these are the results that I've obtained. Does that answer your question, Stephen?
Yes, thank you very much. So yeah, yes. So I wanted to see from like the RNA-seq data, how do I add the column for fold change? Just fold change. Is it possible?
So if you have run a query, let's take a look at that. I think we can do something similar to that here. Let me go back to the query that I just run because I had run a query. Now, the query that you were asked to run in your exercises was a percentile query. But in our group, we were looking at fold change queries. So I have also previously run this fold change query here, which I'm not using as part of my strategy, but is still sitting there as a step. These are the results from my fold change query. I can choose to, and you'll notice that that has the fold change information as one of the columns that was represented here, as well as other information and information that I can then add if I want to know about, well, last time I asked about ortholog groups. And now I can choose to download these results, specify the custom table that I want, and it includes the it includes these query-specific results of fold change that I've run, and I can also choose to download the ortholog group if that's what I want. Now, if I choose to get this, this will be a much longer list because there were more genes here, and maybe I want to customize this further to not show the species name because I already know what the organism is.
And you can also, sorry if you mentioned this, David, already, but it doesn't have to be a fold change or an RNA-seq
Right.
search that you ran. For any list of results that you have in a strategy, you can click on the add columns and you can search for that specific the experiment that you're interested in. So depending on of course, the data, in particular with RNA-seq data or proteomics data, it is mapped to a particular strain, and so you'll see that there are hundreds or tens of RNA-seq data sets, but they may not all be, or they will not all be aligned to your organism of choice. So you'll have to find the one that's actually the specific experiment that was done on, or the data was aligned to your strain of interest.
So that's accurate, but let me also raise one caveat that may be of interest to Stephen. So because Stephen asked specifically about the fold change results, when we look at the fold change query, that generates generic results like the product description, which is a one-to-one mapping for an individual gene, but it also represents query-specific results, such as the fold change data. So this is not an attribute of the gene, it is an attribute of my query. And so when I'm downloading the results of this query, I can get those results. What you may be looking for is the ability to look at the fold change results for these genes, and those are gone, and you can't add them in a column because it is no longer part of this query. So if you wanted to get the fold change information for these genes, you would need to download this information with the fold change results, note down these 8 genes, and obtain those results. Now, you might think that all you want to do is filter all of the query-specific results that you've run in this strategy based on the 8 genes that I'm interested in at the end. And I agree with you that that would be useful, but that would require some development effort. So if you think that that's a high priority, I'd encourage you to click on the Contact Us link and let us know what you'd like to see.
Thank you very much. That's very helpful.
I will point out, for example, that in the breakout group that I was running, we came across a question that we wanted to ask here. That's not what I'm looking for. This is one, let's see, where I had flagged this up. There we are. So there was a question that came up when we were looking at a particular query here for fold change query. We could also choose to run a percentile query as well. But if we've actually run that fold change query, we now can choose to revise that, but no longer have the option to switch to a percentile query. So this is something that should be fairly easy to change, but it would require effort. And because that was a request from users in the group, I entered a query here. I clicked on the Contact Us link and said, users in the ViewPath BB workshop would like to be able to revise a query from fold change to percentile, as you can do in the original selection, but not during the revision. And I attached those images, and that will go into the queue for things to consider as to how much work is involved and how important that is. But lest you think that what seems obvious to you as what you would like to look at must be obvious to us, there are thousands of things that we could possibly do. So your inquiries on the Contact Us link really do make a difference in helping to prioritize where effort gets invested.
Thank you
So
very much.
David, I have a related question. It's pretty much the same when you get the final step is get the auto log, and then once you finish that step, you will not see the fold change of the previous step because that's a different organism. So if I stop at the previous step, I don't go to the last one to find the auto log, but at that time I can download all the genes, right? At that time I can select to show what are the auto logs. So in your table, I will still be able to see all of those fold change,
Right.
the genes, and then it should also list all the auto logs. Is that right?
So in this fold change query, for example, or the percentile query, I can add I think I've already added an ortholog column here. Right. I can download this, and this download will include the fold change results, and I can add other results if I want, what the average is, or it'll include whatever information I want, and I can choose to add the ortholog group if I want.
Right. So this way I would be able to see the autolog genes and also at the same time the fold change.
That's correct. However,
so that'll be very useful.
So you can do that right now, but the confusing aspect of this is that I ran a more complicated search strategy. So I don't want to see all 281 genes. I want to see these 8 genes. And maybe I want to see both the fold change results and the percentile results, which are the result of different queries. So it seems like that should be obvious because you've already run this query and you should be able to download it, but it turns out to be more challenging to implement than you might think. Let me give you an example. Suppose what I had done is I'd run a fold change query and a percentile query, and I had I had chosen to union those results. Now, I will have calculated fold change results for some genes and percentile change for other genes, and some of them will overlap, but not all of them. So since I have not calculated the fold change results for the genes that only were positive in my percentile results, they're not available for me to download at the end. So there are lots of complications here that are more tricky than you might imagine. But as I said before, comments from you are very influential in helping to prioritize effort to solve these strategies. I can't tell you, for example, how many hours went into working on the wording for this query to try to make it clear to identify genes from an upstream region that overlap the exact region of this SNP or overlap within 10 nucleotides of this SNP, or the effort that went in to try to modify these operators here so that if I choose to find genes that are upregulated in any gametocyte sample over every erythrocytic stage sample, as opposed to just the average of those. So a lot of work goes into trying to do this, and your comments really help to prioritize where our effort is directed. But in the meantime, there is the ability. So for what we were trying, what I would do if I wanted to do what you just described, Nir, rather than waiting for weeks or months for the prioritization of that change, I would download these results, download this list of genes, and assemble them using vertical lookups in an Excel table so that I could extract the information that I want. And that's something that I can do in probably less than an hour, as opposed to how long it would take for me to calculate that manually.
Yeah, that's right. Thank you.
Other questions or comments from people?
So maybe I'll answer a question live that came in through the help email from Scott Lanfear, because I still see you on. Hopefully, Scott, I know you had some audio issues earlier. Hopefully you can hear. But the question was about Leishmania mexicana and the ability to find flagellar proteins in Leishmania mexicana. And he was looking, and he couldn't find any. I'm assuming you were doing a go term search, but correct me if I'm wrong. This actually threw me off. That when we first did this, I would look for flagellum. I'm like, that's the first obvious thing I would want to look at if I'm looking at trypanosomes or Giardia or any other flagellated organism, and I would get zero results, which was very frustrating. It turns out the official go term for flagellum is not flagellum, but it's psyllium, right? And so because and so that makes it tricky. So here I just ran the search right as I was doing this. So I looked for L. mexicana. I went to go term. I looked for L. mexicana, which is what you're interested in, and then I typed. If I start typing psyllium, you'll notice that there are a bunch of options, different psyllium go terms. I took this one that's general psyllium and ran this, and that gave me over 1,000 gene results with that go term associated with them. But that actually caused me a lot of frustration when I first was using this. So I'll put this, in case your audio is not working properly, I will put this in an email as well to you. But let me know.
I think it is working. Thanks, Omar. Yeah, I was actually initially trying a text search instead of the Go, but I think I would have run into the same problem.
Let's try psyllium up here and see what happens.
Omar, I did find go terms for flagellum though.
Yeah.
Maybe. Yeah.
I think it's probably in location rather than, it depends on the particular Go term you're looking at. You know, and for Scott, that may or may not have worked using in a text search, depending on the nature of what the annotation is like. You know, you can imagine, for example, if you're doing a search for flagellum and the gene is annotated as flagellar protein,
Yeah.
the term flagellum will not turn up, but flagell star using a wildcard will allow you to pick up that gene. So it's just re-emphasizing the point that Omar made earlier, which is to think of these queries that you're running or these query strategies that you're putting together as an in silico experiment that you'll want to test with both positive and negative controls.
Okay, thanks, David.
So if people are unsure about Go terms, there's a nice site. It's called QuickGo, which also searches for, so you can search for flagellum and psyllium actually comes up. Posting that here. There are synonyms. So Omar,
That's very useful.
Yep.
So as a synonym, the official Go term has flagellum. So Omar, if you take one.
Oh, so what am I supposed to do?
That thing I've posted in the chat, if you go there.
Oh, I see. Apologies. Yeah.
You would have found it there as well. But so if you scroll down, you have the synonym section. And so there's flagellum. And so
I see. Yeah. Okay. Cool. That's a nice site, actually. I've never used this one in particular.
Any other questions that other participants have, or that session facilitators had come up during their breakout groups that might be of general interest? I'll maybe flag up one other that didn't come up actually did come up in my session, but was subject of a note in the chat that I think may have gotten lost in the switch between breakout groups and the public session. There was a question about the interpretation of SNP data, and we'll probably have a chance to do this in the exercises for tomorrow. But I believe you are now seeing my screen with a gene page. Is that correct? Yeah.
Yep.
So the question was, and this is, I think, in Nupur's breakout session, the question was what the rows mean in looking at the SNP polymorphisms. And the rows here mean nothing. For this Plasmodium vivax gene, there are hundreds of thousands of polymorphisms throughout the genome. And if they were all displayed in one line, you wouldn't be able to see them unless you zoomed into the nucleotide level. So looking at the gene level, these are just all arrayed vertically in order to be able to display them all, and not even very visibly unless we were to zoom in quite a bit further. But you can see information on those by clicking on the individual SNPs. It indicates its particular location, the number of strains that were assayed, whether it is a coding or non-SNP. In this case, it's a non-coding SNP. The major allele and the minor allele frequency. In this case, this is a non-coding SNP with a minor allele frequency of 3%. Coding sequence SNPs are indicated in blue, and those in dark blue encode non-synonymous SNPs. So this is a G to C polymorphism where the minor allele would lead to a tryptophan to arginine change, but it is only present in 1% of the samples that were sequenced. And we can go in to take a look at that and see what those strains are and the metadata that's associated with them. This is not necessarily very informative, but it might be depending on what those 1% are. So in my modification, of the query that we ran in the query strategy exercise, I revised my search to look for SNPs that had a minor allele frequency of at least 10%, suggesting that this might be of some functional significance. And again, if we had more metadata, we have robust ability to explore that metadata in the terms here. So we have information for at least some of these on where they were isolated, or their sample source, or the organism that's under investigation. But in other cases, for other species or for other data that you or others may collect, you can imagine if we had information, for example, on the date, as we do, collection year for these samples, or on phenotypic results such as drug sensitivity, you can imagine that this would be hugely useful to be able to search for genes that seem to be under increasing selection between, before and after the introduction of drug.
Great. Well, if there aren't any additional questions or comments, I would suggest that we close the session and give people.
Let me make just
break.
one more comment. Something that Omar had raised earlier. We've been surprised and pleased by the large number of participants in this workshop. We had well over 200 participants today. I'm not sure why the larger participation than previously. It may be because of the large number of emails and publicity and attention that have been surrounding the financial changes in the availability of database resources. And I should say that applies not just to ViewPathDB, but model organism databases as well, which are increasingly concerned about how to ensure that these resources are available to the community. If you'd like to be kept up to date on what is happening, please do take a look under the help list at the frequently asked questions. In addition to questions about data downloading and website tools and gene annotation and various experiments and site usage, we've put a variety of new responses to questions, not only about what ViewPathDB resources are, but about funding status and prospects for the future, which may be of interest to some of you. And we're happy to take questions about it, either at the end of these workshop sessions or by clicking on the Contact Us link if you have other questions to ask. And I think that brings us to the half hour when we're scheduled to close. So with that, why don't we say goodbye and look forward to seeing everyone tomorrow.